The geological timeline marks a period 6 million years ago, indicating specific environmental conditions. Environmental conditions of 6 million years ago include specific climate patterns and geographical distributions. Climate at 6 million years ago can be characterized by average temperatures and precipitation levels. Geographical features at 6 million years ago include the distribution of land masses and major geological formations. Analysis of geological strata provides insights into the environmental conditions of the past. The East African Rift Valley is a series of contiguous geological trenches caused by tectonic forces. Rifting occurs as the lithosphere stretches horizontally, causing the crust to thin and fracture. Volcanic activity is common along the Rift Valley due to magma rising through the thinned crust. The formation of the Rift Valley leads to the creation of diverse habitats, including lakes, mountains, and grasslands. These diverse habitats support a wide range of flora and fauna, contributing to high levels of biodiversity. Climate change induces a gradual shift in global habitats. Dense forests are increasingly transforming into open woodlands due to changing climatic conditions. Continued climate change may further convert woodlands into grasslands. Habitat shifts impact biodiversity and ecosystem stability. These habitat transformations are driven by alterations in temperature and precipitation patterns. The common ape ancestor inhabited a diverse environment of dense forests and open woodlands. This habitat provided a variety of food sources, including fruits, leaves, insects, and small animals. The environment featured both arboreal and terrestrial niches, allowing for diverse forms of locomotion and social behavior. Abundant rainfall and a warm climate supported a rich biodiversity of flora and fauna. The habitat provided ample opportunities for social interaction and the development of complex communication skills. This ancestral environment shaped the physical and behavioral traits of early primates before their evolutionary divergence. Ape and early hominin skeletal structures exhibit key anatomical differences. Ape skulls typically feature a sloping forehead and prominent brow ridges, whereas early hominin skulls show a more vertical forehead and reduced brow ridges. Early hominins possess a more curved spine compared to the straighter spine of apes, facilitating bipedal locomotion. Limb structure differs significantly, with early hominins having shorter arms and longer legs adapted for upright walking, unlike apes. Bipedalism provides a significant advantage in open terrain by improving visibility. Standing upright allows for earlier detection of predators or resources at a distance. Bipedal locomotion is more energy efficient than quadrupedal movement over long distances in open environments. This energy efficiency contributes to increased endurance and range for foraging or migration. Early hominid diets primarily consisted of fruits due to their accessibility and nutritional content. Dietary shifts occurred as hominids expanded into new environments with seasonal fruit availability. Roots and tubers provided a more reliable food source compared to seasonally available fruits. The inclusion of seeds in the diet expanded nutritional diversity, offering essential fats and proteins. A more versatile diet, including roots, tubers, and seeds, supported increased hominid adaptability and survival. Early hominins developed thicker enamel on their teeth as an adaptation to consuming tougher foods. Thicker enamel provided increased resistance to wear and fracture when processing hard and abrasive food items. This dental adaptation allowed early hominins to exploit a wider range of food sources, contributing to their survival and evolution. The development of thicker enamel is a key example of natural selection favoring traits that enhance dietary adaptability. Early hominin brain size was initially comparable to that of apes. The cranial capacity of early hominins began to gradually increase over time. This initial brain enlargement marked a key step in hominin evolution. Comparative analysis reveals a distinct trend of brain size expansion in the hominin lineage. The relative brain size difference between early hominins and apes is a crucial factor in understanding cognitive development. Brain size comparison provides insights into the cognitive capabilities of early hominins. Early hominin groups exhibited social structures distinct from those of contemporary ape societies. Hominin social structures likely involved cooperative behaviors such as collaborative hunting and food gathering. Social bonds within hominin groups were strengthened through shared childcare and mutual protection. The development of early forms of communication facilitated more complex social interactions among hominins. Compared to apes, early hominin social structures may have featured reduced levels of aggression and increased cooperation. The shift towards more complex social organization provided early hominins with a survival advantage. Early stone tools emerged as a crucial innovation for early humans. These tools were instrumental in scavenging meat from animal carcasses. Stone tools aided in processing plant-based foods, expanding dietary options. The use of stone tools provided early humans with a significant survival advantage. Early stone tools represent a key step in technological and cognitive development. Competition for resources is a primary environmental pressure driving evolutionary change. Resource scarcity forces organisms to adapt or face decreased survival rates. Adaptations that improve resource acquisition provide a selective advantage. Competition can lead to the evolution of new traits that reduce direct conflict. Environmental pressure from resource scarcity accelerates the rate of evolutionary change. Evolutionary changes driven by competition and scarcity can result in greater biodiversity. Genetic drift is a mechanism of evolution in which allele frequencies of a population change over generations due to chance. Mutation introduces new genetic variation into a population, providing the raw material for evolution. Genetic drift can lead to the loss of genetic variation within a population, especially in small populations. Mutation can counteract the effects of genetic drift by introducing new alleles, maintaining or increasing genetic diversity. The combined effects of genetic drift and mutation can cause two initially similar lineages to diverge over time. The rate of divergence between lineages is influenced by the population size, mutation rate, and strength of selection pressures. Reproductive isolation prevents gene flow between populations, leading to divergence. Geographic separation is a key factor in initiating reproductive isolation. Different environmental pressures drive natural selection, favoring different traits in isolated populations. Over time, accumulated genetic differences result in reproductive incompatibility. Reproductive isolation can lead to the formation of new species. The separation of hominins from apes was significantly influenced by reproductive isolation. Mosaic evolution describes the differential rates of change in various traits within a lineage. In hominin evolution, some traits evolved rapidly, while others remained relatively unchanged for extended periods. This uneven pace of development results in a mosaic of primitive and derived characteristics in hominin ancestors. For example, bipedalism preceded significant increases in brain size in early hominins. Dental features and jaw structure also exhibited unique evolutionary trajectories within the hominin lineage. Understanding mosaic evolution is crucial for interpreting the fossil record and reconstructing hominin phylogeny. Key fossil discoveries provide evidence for the evolutionary divergence of early hominins. Ardipithecus ramidus is a crucial fossil find that sheds light on early hominin evolution. Fossil evidence helps scientists understand the characteristics and behavior of early hominins. The study of early hominin fossils is essential for tracing the origins of humanity. A phylogenetic tree visually represents the evolutionary relationships between different species. The root of the tree symbolizes the common ape ancestor from which hominins diverged. Branching points on the tree indicate speciation events where populations diverged and evolved into distinct species. The tips of the branches represent extant or extinct hominin species, showcasing the diversity of the hominin lineage. The molecular clock is a technique used to estimate the time of divergence between two species or groups. It relies on the assumption that mutations accumulate at a relatively constant rate over time. By comparing the number of genetic differences between two groups, scientists can estimate how long ago they shared a common ancestor. The molecular clock provides a valuable tool for understanding evolutionary history and the timing of speciation events. Environmental reconstruction aims to depict the habitats of early hominins, including their flora and fauna. The reconstruction includes identifying plant species present in early hominin environments. Reconstruction also requires identifying animal species that coexisted with early hominins. Flora and fauna are integrated to illustrate the food, web, and ecological relationships. The reconstructed environment provides insights into the resources available to early hominins. This reconstruction helps understand the environmental pressures and opportunities that shaped early hominin evolution. Early hominins developed increased cognitive abilities as a behavioral adaptation. Enhanced problem-solving skills were a key aspect of this cognitive development. Improved communication and social interaction also contributed to their survival. These cognitive advancements enabled early hominins to adapt to changing environments. Ape and early hominin hand structures exhibit notable differences in bone length and joint mobility. Apes possess elongated fingers adapted for arboreal locomotion, while early hominins show adaptations for tool manipulation. Increased thumb opposability in early hominins facilitated precision grip, crucial for tool use and fine motor skills. These anatomical differences reflect the evolutionary divergence and adaptation to different ecological niches. The pelvis has evolved to support bipedalism, enabling upright walking and weight distribution. A shorter, broader pelvis provides better stability and balance for bipedal locomotion. Changes in pelvic shape affect the angle of the femur, improving efficiency in bipedal movement. The human pelvis supports larger gluteal muscles, crucial for hip stabilization during walking and running. Evolution of the pelvis represents a key adaptation in the transition from quadrupedal to bipedal movement. The human foot's arch structure is crucial for efficient weight distribution and shock absorption during locomotion. Longitudinal, transverse, and metatarsal arches collectively support the foot, enabling adaptability to varying terrains. The development of arches in the foot is a gradual process, optimizing biomechanical efficiency over time. Toe structure adapts to enhance balance and propulsion during walking and running. The hallux, big toe, plays a significant role in balance and forward propulsion. Changes in toe alignment and flexibility impact overall foot function and gait efficiency. The human spine exhibits natural curves that are essential for maintaining balance and upright posture. Cervical and lumbar regions feature a lordotic curve, inward, while the thoracic and sacral regions exhibit a kyphotic curve, outward. These alternating curves distribute mechanical stress and increase spinal resilience during movement and weight-bearing activities. The spinal curves shift and adapt to support the body's center of gravity, ensuring stability during standing, walking, and other activities. Proper spinal alignment minimizes strain on muscles and ligaments, reducing the risk of back pain and promoting long-term spinal health. Muscle attachments differ significantly between apes and early hominins. These differences reflect adaptations for bipedalism in early hominins. Hominin muscle attachments facilitate efficient upright posture and locomotion. Ape muscle attachments are optimized for quadrupedal movement and climbing. Comparative analysis reveals evolutionary changes in muscle attachments related to bipedal adaptation. Early hominins scavenged for meat by locating carcasses left by predators. Scavenging provided a crucial source of protein and nutrients for hominin survival. Simple stone tools were used to butcher carcasses and access meat and marrow. Scavenging required hominins to compete with other scavengers, such as hyenas and vultures. Social cooperation may have evolved to facilitate group hunting. Group hunting allows for the capture of larger prey, providing more resources for the group. Social cooperation also enhances defense against predators and rival groups. Cooperative defense increases the survival rate of the group, ensuring the continuation of shared genes. Communication development begins with basic vocalizations, such as cries and coos. These early vocalizations gradually evolve into more complex sounds and babbling. Gestures, including pointing and reaching, complement and enhance early vocal communication. The combination of vocalizations and gestures allows for more effective expression of needs and intentions. As language develops, vocalizations become increasingly symbolic and structured. Gestures continue to play a role, supporting and reinforcing verbal communication. Infants exhibit a prolonged period of dependency compared to many other species. This extended dependency necessitates increased parental care and attention. The need for constant feeding, cleaning, and supervision defines infant care. Prolonged infant dependency fosters strong parent-child bonds through consistent interaction and nurturing. Early humans established home bases as central locations for rest, storage, and social interaction. Campsites provided temporary shelter and resource processing during hunting and gathering expeditions. Shelters were constructed from readily available natural materials, such as branches, leaves, and animal hides. The location of home bases and campsites was often determined by access to water, food sources, and natural protection. Early hominin brain development is characterized by an increase in overall size. The cerebral cortex, responsible for higher cognitive functions, exhibits significant expansion. Increased brain folding, or gyrri and sulci, leads to a greater surface area for neural connections. The prefrontal cortex, associated with planning and decision-making, undergoes structural development. These changes collectively contribute to enhanced cognitive abilities in early hominins. Problem-solving involves identifying and analyzing challenges to formulate effective solutions. Learning is the process of acquiring new knowledge, skills, or behaviors through experience, study, or instruction. Cognitive abilities develop through continuous interaction with the environment and active engagement in problem-solving tasks. Effective learning strategies enhance problem-solving skills by providing structured approaches to knowledge acquisition. Cognitive development is a dynamic process that evolves throughout the lifespan, adapting to new challenges and experiences. Stone tool refinement involves techniques to create sharper, more precise edges. Pressure flaking is a method used to carefully remove small flakes for fine detailing. Grinding and polishing can smooth rough surfaces and create specialized shapes. Antler billets and bone tools are used as softer percussors for controlled shaping. Refined stone tools improved hunting efficiency and broadened resource utilization. Evidence suggests early humans controlled fire, marking a significant advancement. Fire provided warmth, protection from predators, and a means to cook food. Cooking with fire improved nutrition by making food easier to digest and killing harmful bacteria. Controlled fire also facilitated social gatherings and the development of early communication. Cooking breaks down complex carbohydrates and proteins, rendering food more digestible. The application of heat during cooking softens plant cell walls, facilitating nutrient release. Cooking inactivates certain toxins and pathogens present in raw foods, enhancing safety. By altering the structure of food, cooking increases the bioavailability of essential nutrients. Early hominins initially inhabited forested environments. A key adaptation was the expansion into more open savanna habitats. Savanna environments presented new challenges and opportunities. This habitat expansion drove evolutionary changes in hominin morphology and behavior. The move into open environments facilitated tool use and cooperative hunting strategies. Animals adapt to seasonal climate changes through migration. Hibernation is a survival strategy for animals during cold winter months. Some animals develop thicker fur or plumage to insulate against the cold. Plants adapt to seasonal changes through dormancy and leaf shedding. Behavioral adaptations include food storage for lean seasons. Physiological changes, such as altered metabolism, aid in surviving seasonal extremes. Effective resource management involves strategic planning to obtain necessary resources. Resource allocation ensures resources are distributed efficiently across different projects or departments. Monitoring resource usage helps in tracking consumption and identifying potential shortages or wastage. Optimization strategies aim to maximize resource utilization and minimize costs. Social learning involves the transmission of knowledge from one individual to another through observation and interaction. This transmission can occur through various mechanisms, including imitation, instruction, and collaboration. Effective social learning requires attention, retention, reproduction, and motivation. Knowledge transmission through social learning enhances individual skills and collective understanding. Cultural evolution begins with the accumulation of knowledge across generations. Each generation builds upon the knowledge of its predecessors, leading to increasingly complex cultural structures. This accumulation fosters advancements in technology, social organization, and artistic expression. The continuous transmission and refinement of knowledge are fundamental to cultural development. DNA comparisons provide genetic evidence supporting evolutionary relationships between apes and humans. High sequence similarity indicates a close evolutionary relationship. Specific gene sequences are analyzed to determine the degree of relatedness. Phylogenetic trees visually represent the evolutionary connections inferred from DNA data. Shared genetic markers provide strong evidence for common ancestry. The fossil record contains gaps due to incomplete preservation and discovery. Fossilization is a rare event, leading to an underrepresentation of past life forms. Geological processes like erosion and tectonic activity can destroy or obscure fossil-bearing strata. Interpreting fossils requires careful analysis of their morphology, context, and geological age. Dating methods, such as radiometric dating, provide a framework for understanding the age of fossils and the sequence of evolutionary events. Phylogenetic analysis uses fossil and modern organism data to reconstruct evolutionary relationships despite the gaps in the record. Pollen analysis is utilized to reconstruct past environments by examining pollen grains preserved in sediment layers. The identification of pollen types reveals the plant species present in a specific area during a particular time. Changes in pollen assemblages across sediment layers indicate shifts in vegetation and environmental conditions over time. Pollen data provides insights into past climate, land use, and human impact on ecosystems. By analyzing pollen, scientists can reconstruct detailed histories of vegetation and environmental change. Isotopic analysis is used to determine the dietary habits of organisms by examining the ratios of stable isotopes in their tissues. Different food sources have distinct isotopic signatures, which are incorporated into the consumer's tissues. By analyzing isotopic ratios in skeletal remains, scientists can infer the geographic origins and migration patterns of past populations. Isotopic analysis provides insights into ecological relationships and environmental changes by tracing the flow of elements through food webs. Comparative genomics identifies key genes by comparing the genomes of different species. Conserved genes across species often indicate functional importance and evolutionary significance. Genes unique to humans may provide insights into traits that distinguish humans from other primates. Identifying these genes can reveal the genetic basis of human-specific traits and evolutionary adaptations. Changes in developmental biology have played a crucial role in human evolution. Alterations in developmental timing, known as heterochrony, can lead to significant morphological differences. Variations in gene regulation during development can result in novel traits and adaptations. Changes in the size and shape of the human brain are partly attributable to developmental modifications. Developmental plasticity allows humans to adapt to diverse environmental conditions. The evolution of bipedalism involves significant developmental changes in skeletal structure and muscle development. Evolutionary trade-offs involve balancing costs and benefits in the development of traits. Increased brain size in humans provided cognitive advantages, but demanded higher energy consumption. Bipedalism freed hands for tool use, but reduced speed and stability compared to quadrupedal locomotion. The development of language facilitated complex communication, but increased vulnerability due to heightened social interaction. Extended lifespan allowed for greater knowledge accumulation, but increased the period of dependency and care. Trade-offs shape human evolution by optimizing resource allocation and adaptation to environmental pressures. Human populations have evolved resistance to diseases through natural selection over generations. Genetic mutations providing resistance to diseases like malaria have become more prevalent in affected populations. The selective pressure exerted by diseases drives the evolution of immunity in human populations. Resistance mechanisms include genetic adaptations that interfere with pathogen entry, replication, or immune evasion. The co-evolutionary arms race between humans and pathogens continues to shape our immune systems. Skin pigmentation is an evolutionary adaptation to varying levels of sunlight exposure across different geographic regions. Melanin, produced by melanocytes, is the primary pigment responsible for skin color and protection against UV radiation. In regions with high sunlight exposure, increased melanin production results in darker skin, providing greater protection against DNA damage. Conversely, in regions with low sunlight exposure, reduced melanin production results in lighter skin, facilitating vitamin D synthesis. The distribution of skin pigmentation across human populations reflects a balance between protection from UV damage and vitamin D synthesis. This adaptation demonstrates the interplay between environmental pressures and genetic variation in shaping human traits. Lactose tolerance evolved as a beneficial adaptation in populations with a history of dairy farming. The ability to digest lactose into adulthood is linked to a genetic mutation that allows continued production of the lactase enzyme. Populations that consume dairy products have a selective advantage due to the nutritional benefits of milk. Lactose tolerance is a prime example of gene culture coevolution, where cultural practices influence genetic selection. High-altitude environments present a challenge due to reduced atmospheric pressure and lower oxygen levels. The human body adapts to high altitude through increased red blood cell production, enhancing oxygen carrying capacity. Increased breathing rate and depth are immediate responses to low oxygen, improving oxygen intake. Pulmonary vasoconstriction redirects blood flow to better oxygenated areas of the lungs, optimizing gas exchange efficiency. Long-term acclimatization involves cellular-level changes to improve oxygen utilization in tissues. Language processing relies on specific brain structures, including Broca's area and Wernicke's area. Broca's area, located in the frontal lobe, is primarily involved in speech production and grammatical processing. Wernicke's area, situated in the temporal lobe, is crucial for language comprehension and meaning. Evolutionarily, these language areas have expanded and become more specialized in humans compared to other primates. The arcuate fasciculus, a bundle of nerve fibers, connects Broca's and Wernicke's areas, facilitating communication between them. Damage to these language-related brain regions can result in various forms of aphasia, affecting speech, comprehension, or both. Symbolic thought involves the use of symbols to represent objects, people, and events not present. Abstract reasoning is the ability to understand and manipulate concepts that are not concrete or directly observable. The development of symbolic thought allows for complex communication and cultural transmission. Abstract reasoning enables problem-solving, planning, and the creation of new ideas. Symbolic thought and abstract reasoning are foundational to higher-level cognitive functions. These cognitive abilities are crucial for learning, adaptation, and navigating complex social environments. Early human societies expressed themselves through art and ritual. Cave paintings served as a form of visual communication and possibly held spiritual significance. Ritualistic behaviors often involved dance, music, and symbolic objects. These early forms of expression reflect humanity's innate desire to create meaning and connection. Complex social structures emerge with the development of governance and hierarchy. Governance establishes rules and order, facilitating cooperation and resource management. Hierarchy defines roles and responsibilities, creating a structured system of authority and influence. Effective governance ensures stability and promotes the collective well-being of the community. Hierarchical structures can optimize resource allocation and task specialization within a society. Early tool use involved simple objects manipulated for basic tasks. Technological advancement led to more complex tools and refined techniques. Modern technology integrates automation and precision in tool-based processes. Continued innovation drives the evolution of tool use and technological capabilities. The agricultural revolution involved the domestication of plants and animals. Plant domestication involved selecting and cultivating wild plants with desirable traits. Animal domestication involved taming and breeding wild animals for human use. Domestication led to increased food production and a more sedentary lifestyle. The agricultural revolution transformed human societies and paved the way for civilization. Urbanization is the process of city development and population concentration. Initial urbanization often begins with the establishment of trade hubs and industrial centers. Technological advancements and economic opportunities drive further urban expansion. Infrastructure development, including transportation and utilities, supports urban growth. Urbanization leads to increased population density and diverse communities. Sustainable urban planning is essential for managing resources and ensuring quality of life in growing cities. Globalization fosters increasing interconnectedness among diverse cultures worldwide. The exchange of ideas, traditions, and technologies accelerates cultural fusion. Digital communication platforms play a crucial role in bridging cultural gaps. This interconnectedness leads to both cultural enrichment and potential homogenization. Human activities significantly impact the environment, leading to various ecological consequences. Deforestation, driven by agriculture and urbanization, reduces biodiversity and disrupts ecosystems. Industrial emissions contribute to air and water pollution, affecting human health and environmental quality. Overconsumption and waste generation lead to resource depletion and environmental degradation. Climate change, driven by greenhouse gas emissions, causes rising temperatures, sea level rise, and extreme weather events. Sustainable practices and conservation efforts can mitigate human impact and promote environmental restoration. Human evolution may continue, driven by genetic mutations and environmental pressures. Technological advancements, such as genetic engineering and artificial intelligence, could significantly alter the course of human development. Environmental changes, including climate change and resource scarcity, may impose selective pressures favoring certain adaptations. Future human evolution could lead to increased cognitive abilities, enhanced physical capabilities, or new forms of social organization. Genetic engineering raises significant ethical considerations regarding its potential impact on human evolution. Ethical debates include concerns about unintended consequences and the potential for unforeseen impacts on future generations. The modification of the human germline raises concerns about the permanence and heritability of genetic changes. Equitable access to genetic engineering technologies is crucial to prevent exacerbating existing social inequalities. Culture significantly influences human evolution by shaping behaviors and social structures. Cultural transmission allows for the accumulation of knowledge across generations, accelerating adaptive changes. Cultural practices can drive the selection of specific genetic traits, leading to gene-culture coevolution. Technological advancements, a product of culture, have fundamentally altered human interactions with the environment. Social norms and institutions, shaped by culture, impact reproductive success and population dynamics. Culture's role in human evolution highlights the interplay between learned behaviors and biological inheritance. Genetic diversity refers to the variety of genes within a species. Cultural diversity encompasses the range of different cultures within a society or region. Genetic diversity enhances a species' ability to adapt to changing environments. Cultural diversity fosters innovation and creativity through the exchange of different ideas and perspectives. Both genetic and cultural diversity are crucial for long-term resilience and sustainability. The study of human origins is an ongoing scientific endeavor. New discoveries continually reshape our understanding of human ancestry. Ongoing research employs advanced technologies to uncover new insights. The narrative of human evolution is subject to continuous refinement and revision. Future explorations promise to reveal even more about our species' past. The story of human origins is a continuing one. A combined timeline illustrates geological and evolutionary events. Geological events, such as the formation of continents and oceans, are shown on the left side of the timeline. Evolutionary events, including the emergence of key species, are displayed on the right side of the timeline. The timeline visually correlates geological changes with significant evolutionary transitions. Key extinction events are highlighted as major turning points in both geological and evolutionary history. The timeline provides a comprehensive overview of Earth's history, linking its physical changes with the development of life. Early hominin fossil discoveries are geographically distributed across specific regions. The map visualizes the spatial distribution of early hominin fossil finds. Fossil distribution patterns provide insights into early hominin migration and evolution. Geographic location is a key factor in understanding the context of hominin fossil discoveries. The distribution map aids in identifying potential areas for future fossil exploration. Comparative skeletal anatomy involves the detailed comparison of bone structures between different species. Apes and hominins exhibit distinct skeletal differences reflecting adaptations to different environments and lifestyles. Cranial capacity and facial structure are key distinguishing features in the skeletal anatomy of apes versus hominins. spinal curvature and pelvic structure reflect differences in posture and bipedal locomotion between apes and hominins. Limb proportions and hand, foot morphology reveal adaptations for arboreal versus terrestrial movement. Bipedal locomotion involves the use of two legs for movement. The process relies on alternating support and swing phases for each leg. Balance is maintained through continuous adjustments of the center of gravity. Muscles in the legs, hips, and core coordinate to produce efficient movement. Variations in speed and terrain alter the mechanics of bipedal locomotion. Climate change simulations model the long-term shifts in temperature and weather patterns. Habitat shift refers to the relocation of species habitats due to changing environmental conditions. Rising temperatures can cause species to migrate towards cooler regions or higher altitudes. Deforestation and urbanization exacerbate habitat loss, further threatening biodiversity. Conservation efforts and sustainable practices can mitigate the impacts of climate change on habitats. Genetic drift refers to random fluctuations in the frequency of a gene variant in a population. These random changes in gene frequency are not driven by natural selection but by chance events. Genetic drift can lead to the loss of some gene variants and the fixation of others, reducing genetic diversity. The effects of genetic drift are more pronounced in small populations. The graph illustrates the evolutionary trend of brain size over time. Brain size generally increases across different species during evolution. The y-axis represents brain size, typically measured in cubic centimeters or relative brain mass. The x-axis represents time, often measured in millions of years ago. Miari. Increases in brain size correlate with enhanced cognitive abilities. The graph provides a visual representation of encephalization, the evolutionary increase in brain size. Dietary habits have undergone significant changes over time. Changes in dietary patterns are often linked to shifts in agricultural practices and food production technologies. Economic factors, such as income levels and food prices, significantly influence dietary choices. Cultural and social influences play a crucial role in shaping dietary preferences and traditions. Public health initiatives and dietary guidelines aim to promote healthier eating habits and prevent diet-related diseases. Reconstructing early hominin social life involves analyzing fossil evidence and environmental data. Fossil analysis provides insights into group size, social hierarchies, and cooperative behaviors. Environmental data reveals information about resource availability and intergroup competition. Reconstructions of early hominin social life are based on interpretations of limited and fragmented evidence. Human evolution involved key adaptations differentiating us from ape ancestors. Bipedalism, or walking upright, is a primary adaptation in human evolution. Increased brain size and complexity mark a significant divergence. Changes in hand structure enabled tool use and manipulation. Dietary shifts and reduced jaw size are notable evolutionary changes. Please subscribe to the channel and hit the notification bell so that you will be updated for more videos.